and we will begin this session on cell matrix interactions with an invited lecture from Dr. Najma Latif from the Yaqub Group.
Okay, good afternoon. So I was invited to give this lecture on adipogenesis and neurogenesis in the normal and tissue-engineered pulmonary valve and the functional implications. So I will talk about our tissue-engineered pulmonary valve and how we've come to this advanced level of remodeling in our valve. So as a background, native valves perform extremely sophisticated functions, and this depends on their specific cellular phenotypes as well as the extracellular matrix composition, which is spatially distributed, and this is tightly regulated by humoral and neuronal factors. And the cell-to-matrix interactions play a crucial role in the valve function, and this cell-matrix interaction translates to enhanced survival and quality of life. We know that none of the available current valve substitutes replicates these functions, and I won't go into the limitations of the current valve substitutes. There is a lot of information about remodeling in tissue-engineered valves. However, the role of adipogenesis and neurogenesis in tissue-engineered pulmonary valves has not been studied, and this is what I will talk about. So the objectives of my talk is to describe the production and functionality of our tissue-engineered pulmonary valve, demonstrate the remodeling process, and then describe the role of adipogenesis and neurogenesis in the normal and that compared to our tissue-engineered pulmonary valve. So we published this paper describing valvulogenesis in a living, innervated pulmonary route. And we use an acellular scaffold, and I will describe how we come about this, how we achieve this remodeling in our acellular scaffold. Before this, we published this paper about anisotropic matrices. So the anisotropic matrix we use is made of polycaprolactone, and we achieve the anisotropy by adjusting the drum rotation speed. There. So by adjusting the drum rotation speed, you can adjust the porosity, the median pore diameter, and the total pore area. So the faster you rotate your drum rotation speed, the higher the porosity, the mean pore diameter, and the total pore area. And the range of fibers that we can achieve through this process, which is a process of jet spraying and not electrospinning, is quite broad. So you can see bundles of fibers. You can see beads, which are hollow. You also find knots. You can have straight fibers, wavy fibers, and branched fibers. And if you look at the distribution of these fibers, I wonder if this will play. So this is a CT scan of our 3D scaffold, and you can see the heterogeneity in the fibers and also the different features inherent in our scaffold, the beads, the thick fibers, the thin fibers, the straight and the wavy fibers. If you look at the distribution of our fibers, we have a very wide distribution. It goes from the nanometer range all the way up to 10 microns, with a mean and median fiber diameter around 0.5 micron. This is significant because when you look at native extracellular matrix proteins, they all fall within the small nanofiber to small microfiber range. So our PCL scaffold is more like native valve tissue extracellular matrix. So this slide shows our valve assembly process. So we use jet spraying to spray 3 sinus molds These are assembled into a holder and a counter. This is then this is an anisotropic layer of PCL scaffold. Then this is jet sprayed with an isotropic layer. We then mount a knitted support, and this is made out of polycaprolactone as well. And this knitted support has various regions of different tension to mimic the native valve regions. This embedded support is then coated with another layer of polycaprolactone, and then these molds are dissolved away to reveal the valve. This is the distal view and the proximal view. So we performed a sheep trial in Germany and Netherlands, and we started with 24 animals. We had 15 successful animals, and we explanted them at weeks of 1, 2, 3, 9, 12, and 26 weeks. So we have a time course of over 6 months of explantation. We performed some in vitro durability testing. So the in vitro durability showed that our acellular scaffold performed 9 million cycles. So 9 million cycles equates to roughly 3 months of time. However, this is in vitro testing. When you implant the scaffold, there is also remodeling going on, so the scaffold doesn't fail at 3 months. In fact, it survives for more than 6 months. The echo data shows good mobility of the leaflets in systole and diastole. We have laminar flow, and we have minimal regurgitation in our valve. This slide shows the in vivo function. So in our tissue-engineered valve, we have preserved root shape. You can still see the sinus shape here. Compared to the control, we have a good size match of the right ventricle. There is no enlargement compared to the control. We also have near normal flow tracings, and MRI shows a double vortex and no abnormal acceleration in our tissue-engineered valve compared to the control. So what actually happens when you implant the scaffold into the sheep? So the first thing to happen within seconds to minutes is that you get protein adsorption from bodily fluids. So this will include albumin, fibrinogen, fibronectin, vitronectin. The fibrinogen is converted to fibrin, and the fibrin binds to the scaffold. So this protein adsorption is what the cells see as soon as it is implanted. It's this biointerface that the circulating cells see. The fibrin binds the cells via integrins and non-integrin receptors. So for leukocytes, they will bind through alpha M and beta 2, and then the endothelial cells and fibroblasts bind through other receptors, alpha V, beta 3, as well as VE cadherin, ICAM, and the selectins. And once the cells are inside the scaffold, they begin to release growth factors and cytokines, such as VEGF, BMPs, TGF-beta, FGF, and PDGF. So as soon as the scaffold is implanted, cells are homing into the scaffold, and by week 1, we have CD45 cells within the scaffold. CD45 cells within the scaffold. Now, these images are all orientated with the luminal side on the right and the abluminal side on the left, so if you bear that in mind. By week 2, we have abundant cells within our scaffold. This is the knit right in the middle of our scaffold. So we have M1 and M2 macrophages. We have a lot of cells expressing alpha SMA, CD31, and we have a lot of fibroblasts. We don't have a marker for fibroblasts, but the fibroblasts are identified through transmission electron microscopy, which we show in our manuscript. By week 3, we have expression of smooth muscle myosin. So the smooth muscle myosin would be within the vasculature. We also have foreign body giant cells, which are these multinucleated cells. And by week 3, we see the beginning of adipocytes and nerve bundles on the outside, on the abluminal side of our scaffold. Between weeks 3 and 6, we see expansion of all the cell types. This slide shows the timing of endothelialization, which begins from 2 weeks and it expands up to 6 months. So in the sinus, you can see the expression of 1 Willebrand factor. In the leaflet, there is reduced expression of 1 Willebrand factor, and it's not a complete endothelial layer. The scanning electron microscopy shows the typical cobblestone morphology of the endothelial cells. We also see endothelial to mesenchymal transition of the endothelial cells, whereby the endothelial cells also express alpha SMA and smooth muscle myosin. We've also looked at the expression of the extracellular matrix over time. So when we look at collagen, it's weakly expressed at week 1, and then you can see the increase at week 2, and more so at week 3. And particularly, you can see it's localized around the knitted support structure. We also see the production of elastin, which is quite difficult in vivo to produce. We see small deposits of elastin, and then by week 2, you can see these small fibers of elastin. And by week 3, you can see elastin in the lamellae of the vasculature. So this slide actually shows the fibrin expression, and not many people show expression of fibrin within valves. So this is a Marcius yellow scarlet blue, and the fibrin is stained red, and the collagen is stained blue. So with as I mentioned, the fibrin adsorbs to the scaffold. So this is the fibrin on the outside, and you see some on the inside. And then it's increasing by week 2. You can see the collagen being produced here on the abluminal side. It peaks at around week 9, and then by week 26, the level of fibrin has decreased. It's a similar situation in the leaflet. So this was the sinus. This is the leaflet. By week 13, you see a maximal expression of fibrin, and by week 26, it seems to it is beginning to resolve, but it hasn't completely resolved as it has in the sinus. So there's still remnants of fibrin in our scaffold by week 26. We showed in our manuscript the expression of extracellular matrix proteins, and we used Picrocereus red in our manuscript, which shows total collagen levels. However, we have now shown that collagen 1, this is a normal sheep sinus. You can see collagen 1 here in the sinus wall. At week 2, we see a low level of collagen 1, and by week 26, you can see the beginnings of aligned collagen in the sinus wall. The leaflet of the pulmonary valve does not show such a marked distribution of or expression of collagen as you would see in a normal aortic valve. You would see a lot of collagen around the fibrosa, but here in the pulmonary valve of the normal sheep, you see more of a diffuse expression. And in the leaflet, by week 26, there is expression, but it seems to be more at the periphery of the leaflets. This is collagen 3. Again, you see aligned collagen in the normal sheep. Weak collagen 3 at week 2, and the beginning of alignment by week 26. A more homogeneous expression of collagen in the leaflet, and the beginnings of collagen 3 in the leaflet, but the leaflet remodeling is a lot slower. This shows the expression of fibronectin, and by week 26, you can see that we have almost equivalent expression of fibronectin as in the normal sheep. The leaflet, this is a normal leaflet showing fibronectin, and the fibronectin level here is almost equivalent in the normal sheep. We've also looked at some proteoglycans. This is versican. So versican in the sinus wall at week 3 and week 25, and in the leaflet, but versican is very low in our leaflet at week 26. Chondroitin sulfate, you can see almost equivalent patterns and levels of expression at week 26, and much weaker expression in the leaflet at week 26. So this slide is a normal section through the normal sheep sinus wall, and it shows the intima, the media, and the adventitia. So the adventitia is a rich compartment of the sinus wall, which contains a lot of adipose tissue. It contains nerve bundles. These are small nerve bundles, vessels, and lots of cells, lots of fibroblasts and macrophages as well. So this is what we would aim for our tissue-engineered valve to look like. So when we look at adipogenesis in our tissue-engineered valve, we see firstly, we see adipose tissue, and we see the expression of leptin at week 3, which is weakly expressed, and we see the expression of fatty acid binding protein 4. By week 26, we see enhanced expression of leptin, but FABP4 is maintained at the same level. When we look at the spatial pattern of expression of the adipose, we can see that at week 2, there is a small depot of adipose tissue towards the distal end of our sinus wall. And these are the small pockets here in this enlarged panel. By week 3, this adipose tissue has expanded. And here you can see the adipose tissue here. And by week 26, it has expanded even more. And there is also a depot of adipose tissue towards the base of our valve. And if you look in this expanded picture, you can see this rich connective tissue. And within this connective tissue is the adipose tissue with these vessels. And these are the nerve bundles within this adventitia. This is another image of our 26-week explanted TEPV. Again, you can see the nerve bundle here, and it's juxtaposed with a very well-defined artery with lamellae stained with elastin and a vein either side. So the expression of nerves and vessels is very closely linked. The nerve bundles were also histologically verified by staining with silver stain. So this is a silver stain showing a nerve bundle. So we looked at the expression of neurogenesis over time in our tissue-engineered pulmonary valve. So this is 2 weeks. At 2 weeks, you can see the beginnings of the expression of neurofilament protein. So neurofilament protein is a structural protein within the nerve bundles. So you see weak expression at 2 weeks, and we can also see the expression of tyrosine hydroxylase. Tyrosine hydroxylase is a sympathetic marker. It's a dopaminergic marker. And by 26 weeks, you can see strong expression for neurofilament proteins showing maturation of the nerve bundles, as well as strong expression of tyrosine hydroxylase here. You can also see that the nerve bundle has grown bigger and matured between weeks 2 and weeks 26. On this side of the slide, you see the expression or lack of expression of substance P. So substance P, there is a nerve bundle right here. We do not see the expression of substance P at weeks 2, but by weeks 26, there is very weak expression in these nerve bundles. There are 3 nerve bundles here, here, and here. And there is the beginning of expression of substance P. Substance P is a sensory nerve marker. And when we look at vasoactive intestinal protein, and this is a parasympathetic marker, we see very little expression as well. There is a nerve bundle right here next to this blood vessel. It's not showing positive at all, but there is a nerve bundle here as well at weeks 26, and this is not being expressed at all. So the majority of the nerve bundles are of the parasympathetic type, and they're expressing tyrosine hydroxylase. This slide shows one of our explanted tissue-engineered valves, and you can see the extent of the adipose tissue here. There is the adipose tissue beginning at it has started for a while at the base of the valve. In some of our valves, we see a complete layer of adventitia with adipose tissue. And if you look in this expanded panel, you can see the connective tissue, there's collagen and elastin. You see these nerve bundles here, and they're juxtaposed with these nerve bundles, with these blood vessels, sorry. So over the course of 26 weeks, we have remodeling in our tissue-engineered pulmonary valve, and we also have an organization of the layering. We have this is a schematic of what is going on, and this is one of what is going on in our valve, and this is an actual explant of our tissue-engineered valve. We have an endothelial layer, we have an internal elastic lamina stained by EVG, elastic Van Giesen. We have a neomedia here, and this would be full of smooth muscle cells. We have an external lamina here, and we have a mature adventitia. The adventitia is full of adipose tissue, and this is white adipose tissue as stained by leptin and FABP4. And within this white adipose tissue, you see vasculogenesis, and you also see neurogenesis. There's a nerve bundle here, and there's many blood vessels in the adventitia. So what are the functional implications of this adipose tissue? There are many functional implications. Its main function is as energy storage and release, and this is through the triglycerides. It also has endocrine functions, such as metabolism and appetite regulation through leptin. It also regulates insulin sensitivity through the expression of adiponectin. It also plays a role in inflammation. It insulates through providing cushions and maintaining body temperature. It also provides protection by cushioning the organ with the neighboring organs. It also has immune function, which is modulated by macrophages. You do find macrophages in the adventitia. And what are the functional implications of neuronal tissue? There are many. The main role is sympathetic activity. Through neurotransmitters such as noradrenaline, it can have effects on smooth muscle cells, which have receptors for noradrenaline and cause constriction. There is also some parasympathetic activity through acetylcholine and vasoactive intestinal protein. These can mediate vasodilation. There is also sensory activity, as shown by weak expression of substance P. And this sensory activity is to detect stretch, injury like pain and inflammation. They maintain vasomotor tone. They can also affect angiogenesis because they release factors that stimulate and guide endothelial cells during healing. They also have an effect on mechanical properties, as the mediators can affect the stiffness of the tissue. They can also play a role in cell growth and collagen synthesis, as shown by the effect of 5-HT on valve interstitial cells. So the adventitia is the most complex compartment of the sinus wall, and it actually houses a variety of cells. The fibroblasts are the most abundant, but you also have macrophages, progenitor cells, vasa vasorum, endothelial cells, and pericytes. The fibroblasts can release a whole host of factors such as VEGF, PDGF, endothelin, transchromin growth factors, fibronectin, thrombin. The The fibroblasts are actually the first cell to migrate to the wound, and they orchestrate this reparative neovascularization. The adventitia is also a niche for stem cells. There are endothelial progenitor cells, mesenchymal stem cells, hematopoietic stem cells, smooth muscle and pericyte progenitors. And under stress and injury, the adventitial cells are often the first to be activated. We see this because we see a high number of smooth muscle alpha actin at week 2 in our scaffold. And this results from activation of the fibroblasts into myofibroblasts and their proliferation. So there is also upregulation of contractile, extracellular matrix, and adhesion proteins through this activation, as well as the secretion of chemokines, cytokines, growth factors, and angiogenic factors. So the adventitia can actually regulate the structure and function of the sinus wall from the outside in. So to conclude, our preliminary data shows an advanced level of rapid remodeling in the sinus wall. This does include elastogenesis, collagen deposition, relevant cell infiltration. We have smooth muscle cells, vasculogenesis. Importantly, we show for the first time that there is neurogenesis and adipogenesis, and it's on the outside of the sinus wall. It's in the adventitia. We are currently addressing the slower remodeling and neurogenesis of the cusp. Thank you very much for your attention. I hope that's okay to put up there.
Yeah, picture. Thank you very much. This presentation is open for discussion. Mike, Caroline?
Dear Nashma, congratulations. A very, very thorough histological overview. We love that because now we see what's going on in the body, right? Really good. I have a question about the other part that you put in the body, which is the PCL. Can you tell a little bit, or the material that you put in, can you tell a little bit about that?
About PCL?
Yeah.
PCL has been used for a long time as a biodegradable, biocompatible material. It's currently used as a suture material, so there are no concerns about its toxicity within the body. It's a long, it's a slow degrading molecule.
Could you see it in the histology? Can you find it back? Or?
You do see it in the histology. You can see it in the early stages where it's not masked by the extracellular matrix proteins. Even, let me see if I can go. Here, in this picture, you can see this is the layer of PCL here, this blue one.
Do you have any idea when we will get rid of it? How long it would take in this case?
It's a slow degrading polymer, so complete degradation we would imagine would be in about 2 years. You can see it's more intact here on the inside of the knitted material, but here it's more degraded on the outside because we have more cells here and there's more cellular activity here.
Okay, thank you so much.
Here is a question.
I think it's a great talk. So general question regarding to all kind of the regenesis in the tissue engineer pulmonary valve. So any kind of control management for such kind of regeneration?
Sorry, I didn't catch
any kind of control for the regeneration. For example, any kind of regeneration, it can be controlled.
Can it be controlled?
Yes.
I'm not sure what that means. Can it be controlled? If you add certain molecules to the scaffold, you can help the regenerative process. You can aid the infiltration of cells, but at the moment we don't want to use any biological molecules to help the regenerative process. But it can be controlled, yes. But we want the body to act as the bioreactor and the body to do all the remodeling. We don't really want to put any biologics into our scaffold.
I got it. So if I understand correctly, you just want to implement your valve into the animal body, let the naturally control.
Yes.
Good, thank you.
All right, I have a question. The knit scaffold, is the objective for that to be degradable as well? And if so, I was curious about how the degradation of that part, I mean, it seems the PCL is staying longer. I can't tell if, you know, how that's persisting, but the relationship with the collagen is interesting. I don't know if you comment on that part.
There's a lot I can comment about on that. The knit is, you can see it over here.
Yeah.
And the cells, wherever they are homing in from, they do seem to really like the knit, because you see there's abundant collagen here around the knit, and you can see these bridges going through the knitted scaffold. So for some reason, the cells really like the topography of the knitted scaffold. The knitted scaffold was meant to be a support structure, but it actually aids the remodeling of our sinus wall.
So it persists then? Is that the idea?
It's the knit. The knit is still there. I know it's a little broken on this slide. It's still there at 6 months, but over the course of 2 years, we anticipate that it will degrade, and it will be taken over by extracellular matrix.
Okay, thank you. All right, if there's no further questions, let's move on, and thank you for the speaker, and we'll go to the next presentation.
Yeah, so our next speaker will be Hong-Go Kim, that will talk about optimal decellularization and xenoantigen removal for tissue valve.
This one.
Good afternoon, ladies and gentlemen. I'm Hong-Go Kim from Seoul National University Hospital. I'm going to present our topic, titled with optimal decellularization and xenoantigen removal for tissue valve. This research was supported by SNUH Ligoni Child Cancer and Rare Disease Project. Tissue valve has been used largely for immune response to antigenic components of the xenogenic tissues, resulting in deterioration, inflammation, and calcification. The major xenoantigens are the alpha-Gal epitope, which is responsible for hyperacute graft rejection on transplant in humans, and the non-human sialic acid and glycoryl uramic acid, which cause acute graft rejection. The aim of this study was to evaluate the safety and efficacy for optimal decellularization and enzymatic treatment for removal of alpha-Gal and non-Gal xenoantigens to reduce the immunogenicity and enhance the biocompatibility of glutarate fixed cardiovascular xenografts. The cardiac xenografts are conventionally cross-linked with glutaraldehyde to have tissue stability and reduce antigenicity. However, the cross-linked xenografts are prone to calcification after long-term implantation in human because of phospholipid and residual unbound aldehyde groups of glutaraldehyde and immune reaction. At the first, our anticonceptive treatment was organic solvent to remove phospholipid, and bind to hydrophobic residues within collagen and elastin. And second anti-calcification protocol is detoxification process to neutralize pre-aldehyde groups forming ship base. And third anti-oxidant treatment is the decellularization to remove major immunogenic cellular components. And the fourth anti-calcification treatment is interstitial space filler to react pre-aldehyde group to inactivate and exclude block, form of area, exclude and block of calcification site. And the role of immune reaction is very important. And alpha-gal and anti-gal immune reaction resulting in chronic rejection, inflammation, and calcification. Alpha-gal epitopes are still present in commercial bioprocess heart valve, even for decellularization. And we proved that the anti-alpha-gal antibody response increased after xenogenic heart valve implantation. So we applied recombinant bacteroides tau micron alpha-gal stage to effectively remove alpha-gal epitopes still expressed in decellularized xenograft. We proved the in vitro study with the lectin histochemistry and the mild-top method and in vivo alpha-gal knockout mouse. And then this 5 treatment protocol was applied in pulmonary stent valve 10 years ago. And then this protocol was applied in patients, more than 700 cases onto this protocol. Now I'm going to talk about the efficacy and safety of peptide and glycostage F treatment for non-gal xenoantigen removal. Looking at materials and method, cardiac xenografts, porcine pericardia, were decellularized with hypotonic buffered solution with SDS and Tritonex, and isotonic solution with enzymatic treatment of alpha-galactose stage for alpha-gal xenoantigen removal, and peptide and glycostage F for non-gal xenoantigen removal, and hypotonic buffered solution and treat with space filler, and then cross-link with glutaraldehyde in organic solvent and detoxify. Materials and method included histology, DNA contents, and SAM, and biomechanical property, and lectin histochemistry, including MRI, WJ, ICA, GSL, ECA, PNA, SBA, WFA, DSL. This lectin histochemistry is representative for xenoantigen expression. After our decellularization, porcine, the HNE staining showed complete decellularization. Without histological change. And the DAPI staining showed the fluorescence signal was completely removed and the DNA content was significantly reduced. Methylene striacrine staining was performed for collagen fiber integrity, and after decellularization, collagen fiber deposition was intact, and the percentage of collagen deposition was not decreased after decellularization. Toluidine blue staging was performed for GAG, and aminoglycan was not affected with our decellularization, and even if alpha-galactose stage was treated, and GAG percentage was not decreased. This is alpha-gal epitope IH staining, shows that this cell decreased alpha-gal epitope expression, and alpha galactose was treated, was added, and then the significantly more reduced alpha-gal epitope expression. And then PNGF treat. I'm going to talk about safety of PNGF treatment. H&E staining showed that there was no histologic change in H&E staining, even if the concentration was increased. And same demonstrated also there's no histological change, even if the PNGF treatment concentration was increased. Looking at biochemical property, tensile stress and tensile displacement, the tensile strain is not changed, not different, even if the PNGF concentration was increased. The permeability test was performed, and there was no difference of permeability, even if PNGF concentration was increased. And looking lectin histochemistry, the Jacqueline, Marai, WJ, ICA, GSL, fluorescence signal was not decreased as with the concentration dependent manner. And also ECA, PNA, SBA, WFA, DSL, also the fluorescence signal decreased with the concentration dependent manner. We performed the quantitative analysis with ELISA, WJ, ECA, JSL, ICA, Marai, WFA, DSL, lectin binding to soluble proteins was decreased as the PNGF concentration increased. And the decel was applied. And after decellularization, the fluorescence signal was decreased, but still remained. And the PNGF was treated, added, and then completely removed. But there was no there was no difference, even if the concentration was increased. This result demonstrates the synergistic effect of decellularization and PNGF treatment. And I'm going to talk about the synergistic effect of alpha-galactose and PNGF treatment. Looking at biochemical properties for general antigen removal, tensile stress, tensile displacement, and permeability was not different, even if PNGF treatment was increased, concentration was increased, and alpha-galactose treatment, the concentration was increased, and PNGF and alpha-galactose treatment was treated together. This lectin histochemistry slide showed the synergistic effect of decel, alpha-galactose treatment, and PNGF treatment. Decel decreased fluorescent signal, but still remained, and the PNGF treatment with PNGF treatment decreased fluorescent signal with concentration dependent manner. Also, alpha-galactose also decreased lectin expression with concentration dependent manner, and the PNGF treatment is much better than alpha-galactose treatment. And decel and alpha-galactose PNGF treatment was treated together, significantly reduced the lectin expression was significantly reduced. So we can see the triple synergistic effect of decel and alpha-galactose and PNGF treatment for general antigen removal. And we performed the ELISA study for quantitative analysis. WJ MARI lectin binding decreased as the PNGF treatment, PNGF concentration increased, and also alpha-galactosis treatment increased. And the PNGF efficacy was much better, and PNGF and alpha-galactosis treated together, the significantly the lectin binding was decreased. WJ, MARI, ECA, ICA1, GSL, WFA, DSL, the same thing. Looking at conclusion, this study demonstrates the safety of our decellularization and general antigen removal. Our decellularization protocols and enzymatic treatment with alpha-galactosidase and PNGF effectively removed the carbohydrates associated genome immunogenicity. These effects may require preclinical studies to confirm enhanced graft biocompatibility and further modification of the tissue engineering protocols in the future. Thank you for good attention.
Questions from the audience? No? Okay, then I have a question, which is mostly a curiosity, at least for me. So I was wondering if your protocol have any observed or anticipated effect on reducing the calcification potential in these xenografts?
We already in vivo efficacy was proved in the alpha-gal mouse in alpha-galactosis treated the xenograft, but PNGF treated graft was not yet. We are going to preclinical study for side effect, anti-calcification for PNGF treatment.
Okay, thank you. Okay, thanks again, our speaker, and we can move next.
Yes, thank you. The next presentation is by Ignacio. Is he here? Yeah, there we go. Excellent.
Good afternoon, everyone. It's a pleasure to be here at Art Valve Society Conference. My name is Ignacio Niosi. I'm a biomedical engineer, and I'm also a PhD student, currently enrolled in a PhD program in biofabrication and tissue engineering fabrication. My scholarship is funded by the Fondazione Rimed. I'm from University of Palermo, and I work within the ERC biomitral project. I would like to start talking about the mitral valve regurgitation that, due to multiple diseases, could cause ultrastructural degeneration of the valve, leading to the need of a surgical substitution. In the field of clinical and preclinical scenario, different options are available, starting from mechanical valves, passing through bioprosthetic valves, and arriving to the frontiers that is represented by the tissue engineered heart valves. However, neither of these category were and are currently designed properly for the mitral valve. For example, because they fail in growth patient compliance and because they lack in the biomechanics mimicking. Our group focuses on biomimicry in the cardiovascular field, and biomitral born to develop a functional microscopically and microscopically biomimetic device made out of polymer, a biodegradable polymer, in order to have an acellular approach, in order to induce, once implanted, the own tissue regeneration of the patients or of the animal. Currently, the heart valve scaffold fabrication, regarding the polymeric one, the field is represented by 3D printing and casting molding, for example, but they are not able to reach and replicate the extracellular matrix-like microstructure. For this reason, techniques such as fiber spinning and electrodeposition emerged, and they represent, in the niche of tissue engineering polymeric valve, the gold standard for pulmonary and aortic valves. However, they fail for mitral valve and tricuspid valve fabrication because mainly of the asymmetric properties of the mitral and tricuspid valve. Fiber spinning, electrowriting, and also electrospinning. However, we believe in electrospinning, and we focused and we spend our efforts on these techniques because it's one of the most cost-effectively heart valves fabrication technique that could be maybe in the future applied in the clinical and could be scaled. For this reason, in order to overcome the electrospinning classical setup, we introduced the double component deposition technology, where the classical full metallic collector is combined with different parts made of metal or plastic. So we will have some parts that will be conductive and other parts that will not be conductive, but that could be polarized. And we designed different double component deposition collectors, starting from the mitral valve shaping through the use of a golden ratio parameterization, passing through a CAD model that we use for the actual fabrication design of the different collectors. Specifically, each collector was characterized by different parameters, such as the surface ratio or the volume ratio, and we used the full metallic collector as a control. We were focused on the study in silico of exploiting in COMSOL some electrostatic simulation in order to analyze the effect of the electrical field distribution on the different collectors and how it affects following the polymer deposition. Why this? Because the electrostatic physics is one of the I would say the most impactful in the electrospinning process. And moreover, in the bottom row, you can see the surface electrical field values histogram, so the distribution of the electrical field on the surface of the different collector, and we quantify the electrical field homogeneity on the surface using a parameter, extrapolating a parameter that is called the full width Adolph centroid, so that represents the width of the electrical field value distribution for the different configuration. After that, we validate this numerical framework with some experimental experiments, and we just fabricated with polycarbonate to ureurethane PCl 12% 9:1 ratio, different anterior leaflets. And why just the anterior leaflets? Because the anterior leaflets represent the critical zone of for the fabrication because of the concavity. And we highlighted and we found that the double component type 2 was the best in terms of thickness homogeneity. After that, we moved towards the study of the setup in order to have an anisotropic leaflet, and we ran some, again, FIM aerodynamic simulation to understand how the air, actually, and pressure fields affect the deposition. And we proceed with the eccentric setup, so we moved the axis of rotation of the collector with having an eccentric configuration, if compared to the non-eccentric configuration where the axis of rotation is passed to the center of the mass of the collector. You can see from the pictures on the bottom that the streamlines became more aligned, and also the magnitude of these streamlines increased in values, and also the pressure gradient became more homogeneous. After that, I would like to to give you an overview about the multi-stages fabrication to have the overall valve. So we have 3 stages each for each particular region of the collector. So the stage 1 for the anterior, the stage 2 for the posterior and for the anterior, and the stage 3 for the annulus. And I would like to highlight how in the stage 3 we introduced this electrode plate in the needle to focus the polymer jet in combination with the introduction of a bottom electrode in the annulus region to increase the electrical field in specific region of the target collector. This is an overview of the collector that was manufactured in 3 different sizes according to some references literature of commissural distances in ships. This is a detailed picture of the collector, and this is an overview of the fabrication outcome for each stage. And I would like to stress about the stage 3, introducing the plate, that allow us to obtain some part of the valve that will be more thick than the other part of the valve, reaching thickness up to 1.5 2 millimeters, mimicking what a common Dacron ring does. This is actually the first valve fabricated, where you can see the thickness distribution along the anterior leaflet, the posterior leaflet, and then the annulus. And I would like to highlight how this thickness distribution could be tuned, varying the size of the collector, but also varying the time in order to reach physiological thickness values, but also over physiological. As mentioned before, we know that the thickness changes, changing the age of the patient. After that, we move towards the anisotropic characterization. In this regards, you can appreciate the structural anisotropy. So we processed some SAM pictures, obtaining the orientation index value, and we compared this parameter with the one obtained from native human fresh samples from the native tissue characterization team. And I would like to let you know that the 0.5 value corresponds to a fully random isotropic tissue, while 1 corresponds to a fully anisotropic tissue. After that, we characterized the anterior leaflet because it's the most important, the moving leaflet of the mitral, also from a functional point of view, through an equistressed Lagrangian biaxial test, and we reported the curves related to the radial and longitudinal direction. We overlapped them with the ones obtained for the native human fresh samples. We can see how along the circumferential direction the curves mostly overlap, while along the radial circumferential they not. That means that along that circumferential direction we were good in aligning the fibers, but along the radial direction this matching in shape is particular complex to reach, because the behavior of the native curve, this J shape, is mainly correlated to the uncrimping of the collagen, and it's because of the hierarchical structure of the collagen, and we don't have any hierarchical structure in our fibers. From the biaxial curves, we extrapolated some parameters, specifically the low initial modulus, calculated in a range between 1 and 1.05 stretch, and also the anisotropic ratio calculated at 400 kilopascal stress value. And we can say, as actually was suggested by the curves themselves, that along the radial direction we have the differences between the radial, the native and the engineered valve. And this is. And also I would like to highlight how the wide distribution for the native tissue, especially regarding the circumferential direction and along and for the anisotropic ratio could be related to the fact that the samples we obtained, yes, were fresh, but they belong to some diseased patients. And we didn't characterize them, dividing them for disease, let's say. So, conclusion, we can say that we were able to obtain homogeneous thickness distribution along the anterior leaflet, that is the most critical leaflet in the mitral valve, thanks to the use of a double component deposition technology. We identified the best setup then for mitral valve anisotropic fabrication through electrostatic and aerodynamic simulation, FEM numerical simulation. And we can say that our valve closely mimic fresh human leaflets anisotropy from architectural and functional point of view. The next steps will be then focused on the hemodynamic assessment. We already are on it, but the data were not robust enough to be shown today. And once we will combine these valves with a set of corde that a colleague of mine is working on, we will assemble, having a mimetic device, so a valve plus a set of corde that will be implanted in an animal sheep model for up to 6 months, starting from September 2025. So, do you have any question?
All right, this presentation is open for discussion. Are there questions? I have a question. So the data you present, and you alluded to a small amount just recently about the variation in the properties that you notice with the native tissue. Now, I certainly understand that there could be differences in, underlying disease conditions, et cetera. But I wonder if you'd comment on the necessity for precision. Like, so for example, in your electrospun scaffolds, you have an extremely tight, range of the properties that you generate, and then the native biology appears to have a wider variation. And certainly, we could appreciate that maybe there's a very tight control locally all over the valve, which has a net variation. But at the same time, there could be other factors that contribute to variation, whether it's the size of the conduit relative to the ventricle or some combinations of things. I wonder, has that been a feature you guys have considered in terms of like trying to maintain such a tight control versus that? Yeah.
Theoretically, yes. Practically, it's more complex to achieve this. By the way, the most important thing is to have a valve that is functional as the native one, in terms of course, hemodynamic parameters in the short term, while allow a certain regeneration that we still don't know, we actually don't know right now. And at a certain point, the cells will colonize the scaffold and will distribute along it according to the stress distribution, because thanks to a mechanotransduction strain driven phenomenon. I mean, we believe that the cells at a certain point will spread and will deposit collagen in certain macroscopic direction according to the stress distribution. So through a mechanotransduction phenomenon.
Okay. Thank you. Okay, well, thank you very much, and we'll move on.
Last speaker of this session, so Saima Imran. He will talk about new directions for tissue manipulating protocol optimization in bioprosthetic heart substitutes.
Thank you very much to provide me opportunity to talk about some of the part of the research which is going on in our group. And actually at this moment, I just am going to talk a little part of the ongoing project which we are working in our group. And this is actually about the tissue manipulation of the for the bioprosthetic substitute, and this is for specifically for the xenoantigen quantification and their enzymatic treatment. And before my presentation, the second last presentation was about the alpha-gal and New5GC expression, and almost the same background of this study which we are doing in our lab. And but at this moment we are I'm discussing and focusing on the commercially available bioprosthetic valves, and we selected randomly depend on the availability in our clinics which were not been used for the clinical implication. So we collected the data, which is very limited, but we collected on the basis of their availability. And actually, this is also the part of our group study which we perform in, sorry. So which we performed in few times ago, and we published about a cohort study regarding the immunogenetic aspect of the commercially available bioprosthetic, and their study with a high number of patients, and it was for 43 months long. So at this moment, we know that the cardiac diseases are a leading cause of mortality, and there are millions of deaths in year 2021, which is 1/3 part of global deaths based on the cardiovascular diseases. And specifically, if we talk about the heart valve diseases, they are affecting 5 to 10% of people which are between the age of 65 and 74 years. And they are affecting 10 to 20% people over the age of 75. And these diseases are actually, included rheumatic heart diseases, aortic valve, stenosis, mitral insufficiency, aortic insufficiency, tricuspid insufficiency, etc. So if we talk about the valve surgery cases, there are 300,000 valve replacement annually, and they are worldwide projected for 2050, which is 850,000. And there are two options available actually with bioprosthetic and mechanical valves. And both of them have definitely their benefits, also with the limitation. So there is anticoagulation, hemodynamic problem, and their durability. So if we are talking about here the durability, then we see that there is a huge problem of these implantation, that the structural valve degeneration is started to happen, and we were discussing in this on this occasion as well about this. And there are 90% of patients which are greater than 65, they do not develop this within first 10 years. And 100% of patients they develop which are less than 35, they develop within 5 years. So the main causes of this degenerative situation is mainly due to the calcification, and also there are mechanical stress and immunological factors which are involved. So here we are more focusing on the immunological factors, because they are related from they are obtained from the animal resources. And I don't know what is happening with this, or I'm doing something wrong. Okay. So these xenoantigen, which we have to focus are the alpha-gal, galactose alpha-1,3 galactose, alpha-gal and Nu5GC, and also SD antigen, but we are more focusing on alpha-gal and Nu5GC. So this study which I was talking before is TransLink cohort study which is published in 2022, and the number of data is quite higher, which is 1,668, and 43 months of this follow-up. After this, this study, it was really found that the preformed antibodies, they bound to the implants, and the complement activation started to happen, inflammatory responses, and endothelial activation as well. So these intravascular thrombosis cause these degenerative phenomena. After this, we are still continue with some of the research. Another part of the work which in the past we have published by Filippo Naso in 2014, in which we published the quantification in the numbers of these alpha-gal antigens in the bioprosthetic valves. In this current study, we got actually 3 different prostheses from Avalus, from Medtronics, Parsival, and the Resilia one, which is the recent one, I think, which we got. We were not very high in number for these samples because these are the samples which were not using in clinics. So we just took them and we used them for this analysis. And basically, we were known about the treatment or pre-treatment of these bioprocesses, in which Medtronic is basically glutaldehyde fixed, and Parsival, they have, and they claim that there is a treatment which is the solution which has adequate phospholipid reduction and aldehyde neutralization. So it is claimed that this Parsival is actually improving the, or it is reducing the chances of calcification than the glutaldehyde. And the third one, which is Resilia from Edwards, and we found that this was in the dry form, and it is actually coated with the glycerols. So the actual treatment we don't know because the companies can tell us better. But we took these 3 different samples, and with this group we found 3 from the Medtronics, 2 from the core, and only one sample we found from two samples from Resilia. We use actually left ventricle native from porcine, which was used as positive control, and also the decellularized one. So after this grouping, we performed 2 types of studies or 2 types of analysis. One is quantification by using ELISA, and for alpha-gal we use non-competitive ELISA, and for the other analysis, which is qualitative, we use immunofluorescence. For the immunofluorescent data, actually the glutaldehyde always cap or preserve the, it hide the presence of alpha-gal, and in real time, in all of the group, we were not very sure about the presence or signals of these alpha-gal. By the way, I was using lectin for the identification. So we did not find lectin binding very efficiently compared to the control, which is myocardia, native myocardia. And with this, we saw the number of, or the distribution of these alpha-gal on the surface from the porcine. While we don't have a lot of signals, but when we treat them with the enzyme, which was from a green green coffee beans. And this is very cheaper source of enzyme of alpha-galactosidase. And we found that it was a bit of removal of the signals we found. But when we quantify them, we found the phenomena that yes, by just removing the glutaraldehyde by simply washing with our buffer system, and there are two different concentration of these this enzyme which we were using. We found that it is really effectively removing the alpha-gal. But this is a part of a study which, a part of samples which we have and which we found, but the qualitative data is not directly matching the qualitative data is not matching directly with the quantitative one. So we rely more on the quantification of the alpha-gal. And with these three samples, we found that three of them, they are having, to some extent, a reduction of the alpha-gal. So for this, for the first Metatronic one, we found this result. And the other one from Parsival, we found two samples, and we saw that the signal were pretty much higher lectin signals, and they were reducing after the treatment with the enzyme. And we saw actually the same phenomena in the ELISA results. Then, in form of Resilia was really resilient. I would say that it was not very good news by removing, by the use of the same range of concentration of the enzyme which we use for the other samples. And we found that after the treatment with the enzyme, we did not see a very, very significant reduction. But on the other hand, if we quantified them by the ELISA, it was showing a reduction because then in the number of the alpha-gal, it can be shown quantitatively that it is reducing. But when we see the quality of signal, we did not found very, very significant result. For me, or from the literature, what we try to find that these preservative or these pre-treatment to remove the, to cope these immunogens from the surface, actually some of them are affecting or some of them are really affecting like an agonist. They are preserving the alpha-gal on the surface, and they are very stubborn to be removed. One more thing is that this alpha-gal is this much stubborn that if we treat the samples, even we have been working also with the animal-based or porcine fresh tissues from different cardiac samples. We found that after treatment on high temperature, ultra low temperature, or normal temperature for very long period, it is very, very difficult to remove them, and they are not affecting by physical conditions. So this is why we can say that they are staying, because the pre-treatment of these valves are actually enhancing or they are supporting their presence on the surface. We also performed the detection of Nu5GC for all of them, and we did not treated them with the enzyme which is specific for Nu5GC. Rather, we wanted to see the specificity of the galactosidase enzyme on these samples. So this is why we kept Nu5GC at this moment in this data, just to see if the enzyme is non-specific. But we found that enzyme is specifically treating the alpha-gal detection, the alpha-gal immunogens, but not on Nu5GC. So Nu5GC in all the sample were supposed to be the same level, or it's not affected. So actually this conclusion made us to the points that these glutaraldehyde are not effective in masking the xenoantigen. And in Avalus, in case of the Percival, the unknown agonistic effect we found. And because rather we saw that it is decreasing the level, it is actually sustaining or enhancing. And the enzyme treatment and washing steps are not affecting. For Resillia is actually totally without any effect of these enzyme treatment. So alpha-galactosidase treatment is significantly reducing alpha-gal expression. It is in some cases, and it is improving it can improve the longevity of these BHVs. We have different responses of different products, so we have to change the condition for each product when we are doing the pre-treatment. So this was the very preliminary data about the present research, and our group is continuously working also with producing new protocol, and we proposed and published the new decellularization protocol in pericardia and cardiac, pulmonary and aortic valve. And this is free of Triton X100, which was forbidden to use from 2017 for the preclinical use and clinical implications. So we already proposed and published this data for the decellularization, and we are also working on the other hand with the removal or try to find more better option to remove these immunogens from the xenotransplant, so it can be useful for the human or clinical use. So this was all about our research. Thank you so much.
Are there any questions from the audience? Yes.
I'm sorry, at the end you mentioned what you're replacing detergent for Triton X. Could you repeat that, and how much do you know functional difference between Triton X and the new
Again,
chemical?
sorry, the detergent?
Yeah, the new detergent you're using instead of Triton X. Can you repeat that?
About the Triton X100, what I understood. Actually, it was because our group was already published a lot of research regarding the Triton-based protocol. And in 2017, we received that it is not being useful. It should not be used for the commercial products or the clinical use. So they actually, FDA proposed themselves various detergents. We worked on 3 of them, and one we found to be very, very effective, which is also eco-friendly, and it is not as stubborn to remove from the tissue as Triton. So we remove it and we replace it with tergitol. This is tergitol, which because it is published, it is not patent, so I can tell you this very in publicly. And actually it was also, later on it was confirmed by the other group who were performing some experiment in Germany for the pulmonary valves. So our group has a lot of work which is already published, so you can check it. So I would like that we have some collaboration, that we can produce some animal-based conduits for using for clinical purpose. This is what we are doing right now. So tergitol is actually the main game changer for us. Any other question?
Yeah. Okay, well, we're actually at time, and we're heading up to a coffee break. So we'll take that, and I guess we'll all report back for the next session at 3:30.
Thank you.
Okay, thank you very much.